Students, if you are confused about the cholesterol or lipid metabolism and how the cholesterol is synthesized inside the body, then this video will help you to understand the synthesis process of cholesterol, even the structure and function of the cholesterol. And at the same time, we will discuss here the importance of the cholesterol synthesis. So let us understand one by one the cholesterol structure, function, as well as the synthesis process. So students, cholesterol is the major sterol in the animal tissues, and cholesterol is present in the tissues and in the plasma either as a free cholesterol or as a storage form, and combined with the long chain fatty acids as a cholesterol ester. In plasma, both forms are transported into the lipoprotein. Plasma low density lipoprotein is the vehicle of uptake of the cholesterol and cholesterol ester into the many tissues. Free cholesterol is removed from the tissues by plasma high density lipoprotein HDL and transported to the liver, where it is eliminated from the body either unchanged or after conversion to the bile acids in the process known as the reverse cholesterol transport. Now to see the structure of cholesterol. All the steroids have the cyclopentano-phenolphthalein ring systems, and they are also membrane component. They are also, cholesterol is the precursor of the bile salts or bile acids, even also vitamin D and steroid hormones. So this is the structure of the cholesterol, and total 27 carbon atoms is there, and there the ring like the designated A, B, C, as well as cyclopentano ring, that is D, and one hydroxyl group at the third position, which is characterized as the sterol, and the hydroxyl group is beta oriented, projecting above the plane of the ring. Now to see the source of the cholesterol, where it comes or where it occurs. Actually, cholesterol can be both synthesized endogenously and obtained from the diet exogenously. So significant amounts of cholesterol only occur in the meat, egg, milk products, and plants and mushrooms, and contain other sterol, but they are very little amount in cholesterol. The fact that vegetarians survive tells us that our capacity to synthesize cholesterol suffices to cover our need for the cholesterol entirely. So, like the other lipids, cholesterol has the low water solubility, requires special mechanism and vehicles for the transport. In the bloodstream, the cholesterol and triglycerides are also transported as a lipoprotein. Now to see the absorption of the cholesterol, if you see cholesterol absorption in the duodenum and proximal jejunum and western diet, and even also 1 by 4 cholesterol is form of the diet. The bulk comes from the bile, even also cell, also intestinal epithelium. Cholesterol is incorporated into the bile salts, micelles, and absorbed into the mucosal cell. And now there is a specific protein which facilitates the transport of cholesterol into the mucosal cell from the micelles. So inside the mucosal cell, cholesterol is re-esterified by the acyl-CoA, and so it is through the enzyme of acyl transferase, cholesterol acyl transferase, a form of cholesterol esters, and incorporated into the chylomicrons. So this is the structures of the how it is transported, it is absorbed into the body. Now to see the biosynthesis process, there is the de novo synthesis process, very important term. Major sites of liver, adrenal cortex, testes, ovaries, intestine, all nucleated cells can synthesize cholesterol, including the arterial wall. The enzymes involved in the synthesis cholesterol are partly that is located in the endoplasmic reticulum and partly in the cytoplasm. So the requirements of this acetyl-CoA provides all carbon, that is 27 carbon atoms, and derived from the acetyl-CoA, and 18 acetyl-CoA are required here. And here also requiring, that is the reducing equivalent supply of NADH, and 16 molecules of NADH are required. So step 1, the synthesis of HMG-CoA, that is beta hydroxy beta methyl glutaryl CoA. Step 2, formation of mevalonic acid or mevalonate. And step 3, production of the isoprenoid units. And step 4, synthesis of the squalane. And step 5, that is the synthesis of the squalane, from squalane to the cholesterol. So these are the 5 steps which we will elaborately discuss in this lectures. And now to see how this cholesterol is synthesized. As you know that synthesis of beta HMG-CoA, there are also 2 molecules of acetyl-CoA. They are from the acetyl-CoA, and another molecule of acetyl-CoA is then added to produce the HMG-CoA. Now the steps of the cholesterol synthesis are given here. synthesis of HMG-CoA, that is beta hydroxy beta methyl glutaralkyl-CoA. Here the two molecules of acetyl-CoA condenses to form the acetyl-CoA. So two acetyl-CoA they condense to form the acetyl-CoA, and with the help of the molecule is then added one molecule of acetyl-CoA again added there and to form the that is called HMG-CoA. So first of all, acetyl-CoA is formed with the help of thiolase enzyme, and one CoA is reduced here. Now secondly, this acetyl-CoA when combined with another acetyl-CoA to form the HMG-CoA, 3-hydroxy-3-methyl or glutaryl-CoA, that is the HMG-CoA. This cytoplasmic enzyme is involved in the cholesterol synthesis, whereas the mitochondrial HMG-CoA synthase participate in the ketone body formation that we will discuss later when we will see in the after the ketone body formations. Now, second is formation of mevalonic acid or mevalonate. How mevalonate is formed from the HMG-CoA. Now, to see HMG-CoA reductase is an enzyme which helps to transfer or convert from HMG-CoA to the mevalonic acids. Here also 2 NADPase is required. It will reduce to form the 2 NADP and one molecule of CoA is reduced from here. Now in step number 3, production of isoprenoid units, the activated 5-carbon intermediate. So the activation occur through the addition of phosphate group. In these 3 steps, reactions catalyzed by the kinase, mevalonate is converted to 3-phospho 5-pyrophospho mevalonate, which is then decarboxylation forms to form the IPP, that is the isopentyl pyrophosphate. Then it is also isomerized to form the DPP. Then IPP and DPP are 5-carbon of isoprenoid units. So in this way, these 5-carbon of isoprenoid units are also produced. Now in the step number 4, synthesis of squalone. How squalone is formed? Squalone, the forms actually the carbon number is 30. So we already know that the carbon 5 was also synthesized through this the previous steps. So carbon 5 it will then convert to carbon 10, then carbon 15. In this way, carbon 30 will be formed. So this stage is the synthesis of cholesterol starts with the isomerization of isopentyl pyrophosphates IPP to the DPP. Then DPP then condense to IPP to form the 10-carbon intermediate, and then further condense of IPP then form the C15, and in this way it will produce the carbon 30 molecules of the substances. Now this conversion of squalone, after forming the squalone, then it will produce the cholesterol. How it can form? It will form through the cyclization. That means cyclization means the squalone undergoes oxidation to form the squalone epoxide, and cyclase converts its 30 carbon of lanosterol, and it is the first steroid compound synthesis, lanosterol. Then this multi-step process is series of 19 enzymatic reaction, and most important reaction reducing of carbon atom from 30 to 27. Then shift of double bond from carbon 8 to carbon 5, and removal of 2-methyl group from carbon 4 and carbon 1 from the carbon 14. So in this way, this is the diagrammatic representation which we are discussing here to understand very clearly. First of all, acetyl-CoA combined with again acetyl-CoA to form the acetyl-CoA thiolase enzyme. Then HMG-CoA synthesis the enzyme. It will produce HMG-CoA, beta hydroxy beta glutaryl-CoA. Then with the help of reductase enzyme, it will dissociate to form the mevalonic acid, 6-carbon compound. Then it will form the 5-carbon units of isoprenoid unit. Then after that, this 5-carbon unit multiply to form the 10-carbon unit, then 15-carbon. In this way, 30-carbon unit of the squalane is formed, and ultimately cholesterol is formed in the 27-carbon units. So this is the process by which then the de novo process by which the cholesterol is synthesized from the acetyl-CoA. So students, I think you all have clear lectures and this synthesis process, and even also the structure and as well as the function of the cholesterol and the absorption process of cholesterol. So if you feel this video helpful or useful, please like, share and subscribe to our channel, and press on the notification bell so you never miss our upcoming lectures in physiology, biochemistry, biology. If you have any question, any queries, any suggestion, feel free to give some comments in our comment sections. And thank you, and I will see you in the next lecture. Until then, keep learning and stay curious.